Today we are talking more in depth about those checkpoints that we talked about on Friday. We're going to talk briefly about what happens when we ignore those checkpoints. Um, so, checkpoints and loss of control is today. Tomorrow we're going to do a little bit of a review. It's the wrong thing. Wednesday we are doing progress questions in class. And then we are going to see how far y'all get. So if um, you don't finish them on Wednesday, we'll have time to do them on Thursday. And then we will either review or finish up Thursday, Friday. So nothing too crazy. But today we are talking about these checkpoints and loss of control. All right. So get my head on straight. Yes. So we talked about checkpoints. We talked about how there are some at G1, G2, and M phases. We talked about how they are just kind of ways for the cell to regulate to make sure that they have everything they need to get done before they move on to the next phase of the cycle. What we didn't talk about is the signals at those checkpoints. So there are internal and external signals at checkpoints. Internal and external signals at checkpoints. Ooh, I'm gonna run out of space. It says checkpoints. Let's see. Specifically, yes, animal cells. When we're talking about animal cells, they have built-in stop signals that will stop the cell cycle. So at these checkpoints, they have signals that stop them whenever they get there. And the cell cycle doesn't start back up again until they have another signal that overrides that stop signal. So, animal cells generally have built-in stop signals. Generally have built-in stop signals that halt the cell cycle until overridden. That stop the cell cycle until overridden. Like I said, we have three important checkpoints, G1, G2, and M. One of the most important ones is the G1 checkpoint. Um, so I'm just gonna write critical G1 checkpoint. At this G1 checkpoint, if a cell does not receive the go-ahead, so if it goes through if it goes through all of its growing that it does in that G1 phase, right? It goes through all of its growing. It hits that checkpoint and it stops, right? If it doesn't get the signal to go ahead, it will exit the cell cycle as we know it, that G1S, G2M, and enter a special phase called G0, which is where it's like essentially dormant. There's no growth. It'll still function as a cell, but it's done. It's not gonna grow. It's not going to divide. So, critical. How did I make that L into a exclamation point? At the G1 checkpoint, if it does not receive the signal, it will enter a non-dividing state called G0. If a cell does not pass this checkpoint, or does not receive the go-ahead signal, it will enter a non-dividing state called G0. A non-dividing state called G0. Now, there is a way to get out of G0. There's a way to kind of kick up that cycle again, and that is through growth factors. Growth factors are a protein that are released by other cells that stimulate them to divide. So the cells in your skin, right, if you're not actively hurt, they're typically in G0. But if you get a cut, like the paper cut that I filled with hand sanitizer this morning, the cells around that cut will release a protein called growth factor that will stimulate your skin cells to grow and start the healing process. So, to get out of G0, that's what happens. So, let's see. Let's say to get out of G0, to get out of G0, external cues like growth factors. Stimulate division. Ooh, I'm getting real close to the board today. So, and like I said, those growth factors are proteins secreted by other cells. These growth factors are also really prevalent in the process of development. So, going from like zygote to blastocyst to embryo to fetus. To get those cells to grow rapidly, the body is chock full of growth factors. So that is an example of an external kind of stimulant to get cells to pass a checkpoint. There are also internal signal checkpoints. So that one was external, it was coming from another cell. But the cell also makes signaling molecules within itself in order to pass checkpoints as well. So internal signal checkpoints. Internal signal checkpoint. All right, example. And this one is going to be that M phase checkpoint. So there's a checkpoint in M phase, and it is right before anaphase. So right before anaphase. So right before anaphase, our spindle fibers have those kinetochores, right, on the ends of them, and our centromeres, centromeres on our chromosomes have those kinetochores. And only when those kinetochores on the spindle fibers are connected to the kinetochores on the centromeres does a molecular signal get activated. So we wait until all of the chromosomes are connected to spindle fibers, a signal is released, and that will allow the cell to go into anaphase. Because it's not going to start pulling stuff apart until it's connected to everything. That way we don't have chromosomal abnormalities. When this doesn't happen, that's when we can get chromosomal disorders. like Turner syndrome or Down syndrome, stuff like that. So, or just stuff gets too sticky. We'll talk about that next semester. So, only when the kinetochores are attached does the correct molecular signal become active. Only when kinetochores are all attached do the correct regulatory signals, that's not how you spell, get activated. All right. So that's our internal checkpoint example. They have a special cell or a special molecule rather that doesn't become active until all of those kinetochores are properly fastened in their place. Kind of like your car beeps at you dramatically if you don't have your seatbelt plugged in. Plugged in? Clicked? But there are other examples that aren't necessarily cell signals. There are physical regulators as well. So physical signals, if you will, that come from outside the cell. So I got two of those. Physical, external regulation. One of these is density dependent inhibition. Density dependent inhibition. So, if you took a culture of bacteria, right, and you let it grow on a petri dish, right, you gave it all the food, all the water, all the oxygen, all the nutrients it needs. You just keep watering it like a plant, right? It'll grow, it'll grow, it'll grow until eventually it stops. It fills up the whole plate, right? Those cells are too crowded to grow anymore. So they stop. But if you were to take a little swab off that plate, put it onto another plate, it'll keep growing, right? So if cells are too crowded, they'll stop dividing. So cells are too crowded. They will stop dividing. As we heal, right? Eventually, our cells will come back together, and we will need to stop the growth signaling, and that crowding is another way to do that. Another example of like a physical thing that affects cell growth is anchorage dependence. Ooh, I need to stop writing cursive. Anchorage dependence. So some cells will only grow if they are attached to a surface, whether it be like a tissue on our bodies or like the walls of a test tube or a beaker, right? A cell is not going to grow unless it has something that it's held onto because it's not stable enough to make the signals to actually divide. So some cells must be attached to something to divide. Some cells must be attached to something to divide. So those are our cell signals. Talked about how the cell cycle works, why the cell cycle why the cell cycle works. And now we're going to talk about what happens when that cell cycle goes wrong. And the answer to that is cancer. So if a cell blows past these checkpoints, if something happens to where it doesn't make the right regulatory molecule, if something happens to where they don't make the receptors for the right regulatory molecule, something happens to that cell where it just kind of keeps blowing past those checkpoints. That's when we get cancer cells. So loss of cell cycle control. Cancer. There is a leading hypothesis. If we know that cancer cells don't listen to cell regulators, right? Either or we don't, or sorry, my brain's not back yet. So we are led to believe, because we know that cancer cells don't listen to the cell cycle, that cancer cells either don't need their growth factors to grow, they make their own growth factors, or they have an abnormality in a signaling pathway. That means that will activate that signal whether or not the growth factors are there. So if a mutation happens in a signaling pathway to where one of the signaling molecules is always on, like it can't be unactivated, then that cell signal will just always be there. And that's another way that they think that cancer cells could happen. So, hypothesis. We think that cancer cells don't need growth factors. Make their own growth factors. Or they have an abnormality in a signaling pathway that always activates the division signal. Abnormality in a signaling pathway that always conveys growth signals. All right. So that's our hypothesis. We know that the biggest difference between cancer cells and regular cells is that cancer cells don't listen to the cell cycle. That's the biggest difference. But there are some other abnormalities in cancer cells that make them different from regular cells. So, abnormalities beyond disregarding the cell cycle. So, abnormalities apart from cycle control loss. One of them is that so when body cells stop dividing, it's at one of those checkpoints, right? They don't get they don't get the go ahead signal. They'll stop at one of those checkpoints until the checkpoint is met. Cancer cells will just stop randomly. They don't need a signal, or they messed up a signal. Cancer cells will just stop dividing randomly, not necessarily at checkpoints. If cancer cells stop dividing, they do so randomly. Not at checkpoints. So if we were to watch cancer cells under a microscope, and a cell stopped dividing in the middle of metaphase, right? That's not a checkpoint. We have a checkpoint at anaphase, but we don't have a checkpoint in the middle or the beginning of metaphase. So if it were to stop there and just not divide anymore, we could say that that was a sign that this culture was cancer cells instead of regular cells. Another one is essentially immortality. These cells, body cells, eventually wear down, right? They'll eventually stop dividing. But cancer cells will just keep going and keep going and keep going, and as long as they have the stuff that they need to grow, they will keep growing. Have you ever heard about HeLa cells before with Henrietta Lacks? No? Okay, that's what tomorrow will be then. Not to get too into it today, but one of the main reasons we are able to study cancer cells is because a woman named Henrietta Lacks went to a clinic and for cervical cancer, right? But she was a Black woman in a very poor neighborhood and was taken advantage of a lot in this situation. And what happened was the doctors took the sample from her like pap smear and used it to study cancer cells. And these cells had a specific mutation that caused them to keep dividing and keep dividing and keep dividing. We were finally able to culture human cells. So almost every lab in the world has these cells now because it's been decades. And so these immortal cells are also called HeLa cells. Henrietta Lacks. So we'll talk about that a little bit more tomorrow then. But that's another abnormality. And finally, let's see. Yeah, no. So these cells in a culture that have the ability to divide indefinitely are said to have undergone a process called transformation. So cells in a culture that can divide indefinitely are said to have undergone transformation. Transformation is the process that causes cells to behave like cancer cells. The process that causes cells to behave like cancer cells. Whoop. My arms. I'm tired today. All right. So that's cancer cells in culture. That's just like if you were looking at a cancer cell under a microscope, if you were looking at a cancer cell in a biopsy, that's what it would behave like. But in the body, things act a little differently. We can actually study like the actual effects of cancer cells. So that was cancer cells just in general. Now we're going to look a little bit more specifically at cancer cells in the body. In your past biology classes, have you done any work with like cancer checkpoints, causes, and no? Okay. So, cancer cells in the body. What happens is it all starts with one cell. One cell becomes a cancer cell. This could be anything from a genetic mutation caused by inheritance or exposure to some sort of carcinogen, smoking, vaping now, sun damage, radiation, stuff like that. Anything that can cause damage to DNA can cause a cell to mutate in a way that makes it become a cancer cell. Usually, these altered cells have changed the proteins on their cell surface. When you change DNA, odds are you're going to change one of those surface cells. So usually, proteins on the surface of the cell will also change. And this will activate an immune response and it'll just kill the cell. So, if the proteins on the surface of the cell change, the body is able to be like, that ain't me, and kill it off. But if we can't destroy it, that cell will proliferate or just keep growing and keep growing. If that cell proliferates, it will become a tumor. If not destroyed, proliferation, and that will cause tumor formation. Yeah, okay. But there are a couple different types of tumors. A benign tumor remains at its original site. Meaning it's not going to spread. It's not mutated so badly that it can't that it can survive in other parts of the body. So when a cancer cell spreads, it's because that cell was damaged so badly it doesn't matter what type of tissue that it becomes or that it invades. But these benign tumors, goodness gracious, that is not legible. Benign tumors remain at the original site of where they are. So if you are, or if someone is to remove a benign tumor, usually it's just surgery, go in, cut it out, you're done. So if you have a benign tumor, it is not considered cancerous because it's not spreading. When someone has cancer, they have a malignant tumor. Malignant tumors spread to new tissues and disrupt functions. Spread to new tissues and disrupt functions. This is what medicinally or medically, medically speaking, what cancer is. If a cell can spread to another part of the body, it is considered a cancer cell. So not only can they impede or disrupt cellular functions, they can also secrete their own growth signals or their own signaling molecules that cause blood vessels and lymph vessels to grow toward the tumor. So they may also secrete signals that cause lymph and blood vessels to grow toward the tumor. If this happens, That's all. That makes removal a lot more difficult because if it's heavily uh, like connected to the blood and lymph node system, not only um, is it hard to get it out without superfluous bleeding, but also if a few cells get into the bloodstream or the lymphatic system, that allows the cancer to spread to other areas. Um, so if a few cells enter those vessels, they can travel to other parts of the body. This process, the spread of cancer cells, is called metastasis. Metastasis is the spread of cancer cells. Goodness gracious. To other tissues. Or to locations distant from its original site. So when a doctor says that the cancer has spread or the cancer has metastasized, it's the same thing. Studying cancer cells, how they happen, how they behave, how they spread, how they come to be, allows us to get a stronger look into how to treat them. I'm not going to go super in detail here, just hitting the high points. Radiation is both a cause and a treatment for cancer. I know, okay, so a lot of the times they use removal and radiation together, just so if they don't think they got the entire tumor, can go in and radiate the area. My mother got thyroid cancer when I was little, so they went in and they removed the actual tumor, but they also dosed her with high energy radiation. Killed everything, good. But the whole point of high energy radiation is that it damages DNA. That damaged DNA, when it comes to like tanning or like exposed to radiation in the lab, like that causes cancer because it can mess with DNA. But the high energy radiation that they use to treat cancer goes about targeting DNA that's replicating fast. So the faster a DNA molecule is dividing, the more likely it is that radiation is going to mess something up. The point of it is to get that DNA so messed up that the cell can't survive anymore. So it targets the fast replicating DNA. High energy radiation damages DNA faster in cancer cells. And then we also have chemotherapy or chemotherapeutic drugs. The whole point of chemotherapy is to interfere with steps in the cell cycle in specifically fast dividing cells. So chemotherapy stops fast dividing cells by interfering with the cell cycle. This is why people who undergo chemotherapy lose their hair, because the cells in our hair follicles divide a lot faster than any other body cells. So if chemotherapy trigger or attacks fast dividing cells, they'll get the cancer cells because those are fast dividing, but they can't differentiate between cancer cells and hair follicle cells because all they know is fast dividing equals bad. The same thing goes for why people who undergo chemotherapy have really bad stomach issues, because those cells on our linings in our stomachs divide really, really fast. They have to in order to protect us from our stomach acid. So fast dividing cells in stomach and intestinal linings means fast dividing chemotherapy is going to get it. But the pros outweigh the cons. If we can get rid of the cancer cells, stomach and hair follicles can return, and hopefully those cancer cells will not. And then also they can be more targeted, and this is more cutting edge. Let's see. One example is there is a certain type of breast cancer cell that has a specific protein on the cell surface. Not all breast cancers, but some breast cancers give the cell a specific surface protein. Some breast cancer cells have a specific surface protein. And in these cases, the treatments, goodness gracious, for these cancer cells are more specific, meaning we can use the immune system to target those proteins on the surface of the cell, or we can design something that attacks that one surface protein that hopefully doesn't interact with any other cells. And those are just some examples. Day by day, the technology gets better and better, which is a good thing. Does anybody have any questions? No? Okay. That's all I have for you today. I don't have an exit ticket for you either. Got a little busy this weekend. So we have like 5 to 7 minutes to do the vibe. Yeah. Like I said, tomorrow we're gonna kind of review a little bit. I'll probably gear it a little bit more towards Henrietta Lacks just because I like talking about people. I think her story needs to be told.